Picture an intensive care unit. A patient is recovering from a severe burn. They survived the initial thermal trauma and their skin is beginning to heal, but suddenly their blood pressure plummets. They are crashing into lethal septic shock. This collapse stems from an internal structural failure. A specific microscopic molecule has been exhausted, creating a systemic vulnerability that no antibiotic can address. Without this molecule, the invisible protective mucus wall lining the patient's intestines physically dissolves. The bacteria that normally live peacefully inside the gut lumen find themselves staring at a bare epithelial wall, and they march directly across that boundary into the bloodstream. That missing architectural anchor is an essential amino acid called L-Threonine. Human survival relies entirely on an invisible microscopic wall to keep our internal microbiome from digesting us. The structural integrity of that wall depends on this single chemical connector. To understand how our bodies are constructed and maintained, we need to decode threonine's physical chemistry, trace how the body triages it during systemic trauma, and identify how clinical laboratories track its levels to prevent a crisis. We begin with a biological vulnerability. The human genome completely lacks the genetic pathways required to synthesize threonine from scratch. Because it is strictly essential, the body must acquire it entirely through dietary protein, like turkey breast or lentils. This reliance on external intake makes our internal supply chains fragile. Because we cannot manufacture it, the human body must ruthlessly hoard and prioritize every microgram it absorbs to prevent systemic collapse. Threonine possesses a second chiral center with 4 spatial shapes, but human ribosomes strictly accept the 2s3rl enantiomer. Its side chain hydroxyl group acts as a sticky connector. The Golgi targets this connector, forcing complex sugars to bind and trap polar water molecules, forming mucin. This thermodynamic reaction expands the physical volume of the molecule, transforming thin, watery fluids into a thick, protective matrix known as mucin 2 gel. Threonine performs a second parallel function as a high-speed cellular alarm switch. Serine-threonine kinases seek out that exact same hydroxyl connector to send urgent signals. They snap a highly negative phosphate group onto threonine. This massive negative charge creates violent magnetic repulsion, forcing the entire protein structure to instantly twist and flip into an on state. Threonine translates chemical data into real-time physical action. It builds protective walls by binding sugars and flips cellular switches by capturing phosphates. A healthy adult contains 35 grams of threonine, 50% locked in skeletal muscle, 30% concentrated in the gut, and a tiny fraction in plasma. Intestinal cells immediately extract 60% of incoming dietary threonine, constantly consuming it to rebuild mucin gel and synthesize antibodies. It specifically forms the dense, hydrogen-bonded hinges of secretory IgA. Because bacterial pathogens release enzymes that act like molecular scissors to snip our defenses in half, placing threonine at the hinge point ensures the antibody structure resists cleavage. Returning to our crashing ICU patient, severe burn trauma forces the body into a hypercatabolic state. Metabolism goes into overdrive, and the gut barrier rapidly starves for raw materials. The body initiates an extreme emergency response, flooding the system with cortisol to trigger severe proteolysis, actively breaking down skeletal muscle to release vaulted threonine. The vascular system then rushes this newly liberated threonine straight to the splanchnic bed, the network of abdominal organs, to urgently rebuild the collapsing mucus wall. The human organism will literally sacrifice its own biceps to save the foundational integrity of the gut barrier. The demand for this structural molecule begins at birth. Preterm and term neonates rely entirely on threonine-rich colostrum to synthesize their very first intestinal barrier, protecting the newborn gut. In contrast, as we age, a natural decline in mucin production physically thins this barrier. This leaves the elderly vulnerable to mucosal atrophy and low-grade infections. But an excess of this building block is equally dangerous. In a rare condition called hyperthreonemia, plasma levels can spike over the 600 micromoles per liter panic threshold. This occurs when threonine dehydratase, the specific liver enzyme responsible for destroying excess threonine, is genetically deficient. Molecule backs up and floods systemic fluids. This massive flood physically jams the limited transport gates at the blood-brain barrier. It monopolizes the entrance, starving the brain of other vital amino acids needed to build neurotransmitters, which leads directly to metabolic encephalopathy. Baseline health requires walking a narrow tightrope. Falling into deficiency invites lethal sepsis, while tipping into excess poisons the brain. To navigate this tightrope, pathologists rely on liquid chromatography tandem mass spectrometry, or LC-MS-MS. It is the absolute gold standard for tracking this elusive molecule in the clinical laboratory. This schematic demonstrates how a mass spectrometer operates. It vaporizes a separated blood sample into a gas and blasts it through a powerful electromagnetic field. By measuring exactly how the particles deflect in that field, the machine determines the precise mass-to-charge ratio of every single molecule in the sample. Yet this extreme precision is vulnerable to pseudo-hyper-threonemia, a diagnostic illusion that creates a false positive on the lab report. The first cause of this illusion is a postprandial surge. If a patient eats a high-protein meal within a few hours of their blood draw, rapid gut absorption creates a massive 40% false spike on the readout. The patient is perfectly healthy, but the machine reads a toxic high. The second cause is mechanical hemolysis. If a phlebotomist struggles to find a vein and uses too much suction, or if the lab spins the centrifuge too fast, the intense physical pressure damages the red blood cells. Because red blood cells are packed with high concentrations of intracellular threonine, popping them spills that material directly into the plasma matrix. The mass spectrometer measures this spilled cellular content, tricking the machine into reporting a lethal liver failure. Multi-million-dollar diagnostic machinery is entirely useless if the physical realities of the blood draw are compromised. Because of these illusions, modern pathology labs build automated mathematical defenses directly into their information software. This graph illustrates the Delta Check protocol. The system continuously tracks a patient's levels over time. If a patient's plasma concentration jumps by more than 40 micromoles per liter within a 24-hour window, the system automatically triggers a threshold boundary and freezes the report before it reaches the doctor. This alert triggers a manual investigation. The pathologist must inspect the test tube for the pinkish tint of ruptured cells or verify fasting timestamps to ensure the data is valid. To safely manage the molecule's volatility, clinical laboratories must adhere to strict quality control parameters, maintaining a total allowable error of plus or minus 15%. For clinicians treating critical care patients, there is a clear directive. Doctors should not rely on total albumin to assess gut health. A localized threonine starvation will collapse the mucosal barrier long before systemic protein markers ever drop. If a deficiency is caught early, the treatment is remarkably straightforward. Administering 1 to 3 grams a day of oral L-Threonine provides the raw materials needed to rapidly rebuild the mucin architecture. This molecule serves as the foundational anchor of the mucin gel, the uncuttable hinge of our immune defenses, and the physical switch driving cellular communication. The barrier separating human life from lethal sepsis is not a static wall. It is a dynamic, living ecosystem maintained minute by minute by a single, ruthlessly guarded molecule.